Hi guys, Mr. Shea here. Today I'm going to be demonstrating a relatively straightforward experiment where I will be extracting DNA from a fruit. So step one is to create a makeshift DNA extraction buffer. This can be done by simply adding salt and detergent to water. Ensure that you mix the salt into the water until it's completely dissolved. It might help to use warm water here as it has greater solubility than cold water. The salt in this solution will aid in breaking up protein chains that bind around the nucleic acids, and detergent, which I'm adding here now, will help dissolve the phospholipid bilayers of cell membranes and organelles. This extraction buffer will basically help provide us access to the DNA contained within the fruit cells. I've chosen to extract DNA from a banana in this experiment, but you can choose to use any fruit. Blueberries and strawberries work quite well with this method too. Next, you have to grind the fruit and add the DNA buffer. Place your fruit into a Ziploc bag and with your fingers or your fist, grind the fruit for a couple of minutes. Careful not to break the bag. The mechanical force that's applied to the fruit here will aid in breaking open the cell walls and releasing cellular contents, including DNA. Now add the DNA extraction buffer you prepared in the first step. Again, this will help in removing proteins and other cellular components by breaking down cell membranes even more and disrupting lipid-protein interactions. So now you're going to pour the fruit slurry into a filtration apparatus, allowing it to drip for about 5 to 10 minutes. Filtering strains all the large cellular junk and cellular debris out of the mix. The DNA, still tightly wound, is so small it slips straight through the filter paper. This may take a while, and if you're short on time, you may not get much. But don't worry too much because there's a lot of DNA contained within just a drop of this liquid. Now you gotta pour the liquid into a small test tube and add ethanol. From this point onwards, you must be very careful not to agitate the mixture. Adding ethanol causes DNA to precipitate out of the solution in the presence of alcohol due to its lower solubility in alcohol compared to water. The alcohol causes the DNA molecules to become less hydrated and aggregate together, resulting in their precipitation. The tiny bits of wispy junk floating in the alcohol just above the boundary layer is the purified DNA. To collect the DNA, dip a glass rod or a pipette into the tube where the DNA extract is suspended. If the procedure has worked well, which it often doesn't, you will get long strands of DNA forming. Here, you want to gently wind up the precipitated DNA and lift it up out of the test tube and onto a glass slide. Ideally, you have long strands of a mucus-like substance upon which you can place a cover slip and observe under an optical microscope. I had to do this step a number of times before I finally got some strands forming. As we conclude this DNA extraction experiment, it's important to reflect on the broader implications of our findings and how they connect to genetic technologies. By doing this experiment, you've witnessed firsthand the remarkable process of extracting DNA from fruit, a fundamental step in biotechnology and genetic engineering. By mastering techniques like DNA extraction, we lay the groundwork for understanding and manipulating the building blocks of life, from genetically modified crops that resist pests to gene therapies that treat genetic diseases. The applications of genetic technologies are vast and ever expanding.